already talked about pyruvate conversion. So what we're doing is looking at that very first step of glyceraldehyde phosphate and converting that across to the bisphosphate. And one of the big things that we mentioned within this is that we did not use ADP to generate the phosphate, okay, or to put that phosphate in there. We got a free phosphate from solution coming into this, okay, which is a really important aspect to this reaction, because ultimately what that then does is allows us to make ATP later on down the road as a net positive. If we'd used ATP to put on this phosphate, all we'd be doing is going backwards and forwards. No net production of energy. Okay? Other things to address within this is that it's doing a heck of a lot of chemistry. Okay? It's kind of neat. And we will keep seeing this type of enzyme pop up. We are doing both an oxidation. Okay, so if we look at what's changing, we're doing an oxidation about that carbon, but further we're now also putting on the phosphate. So we're doing both things. So if we're trying to address which of these things happen first, well, to put the phosphate on, we have to have something attack the phosphate. Okay, our phosphate has that phosphorus in the middle, which is a really good electrophile, which means our glyceraldehyde 3-phosphate needs to act as a nucleophile. And in its current state, it is not a good nucleophile. Okay, so the first thing that has to happen is the oxidation to get that to the carboxylic acid. Once we're at the carboxylic acid, we have a reasonable nucleophile. That nucleophile can then attack our phosphate. Okay, so we have to run the oxidative process first. And that's going to be facilitated by our NAD+, helping to shift hydrogens around, or technically hydrides around through that process. Not the easiest thing to go through and do. We're not just going to see water come in and attack this to give us the acid. Water's not a strong enough nucleophile, and that's not a strong enough electrophile for that initial oxidative step. So what ends up happening is that we use a much stronger nucleophile in a sulfur group. So if you think back to what we talked about with our coenzyme A, we mentioned that it had that thiol group at the end of it, and that thiol was really useful as a good nucleophile. Well, guess what cysteine supplies? If you look at its amino acid structure, it is also a thiol with another SH, okay, a terminal sulfur hydrogen bond.
Can you hold the screen down?
Sure. Sorry. That sulfur is what's going to act initially as our nucleophile in here to activate that carbon. And by activating, what we end up with is now a single bond out to this oxygen, and then our cysteine residue. Okay. That carbon is now much more active, much more inclined to react with water. Okay. When it reacts with water, we can also, not with water. First thing that happens is our negative oxygen will come back down. I know that looks like a 1. Let me try that. With the sideways iPad, it looked like a negative. The negative can come back down and kick out the hydride out to NAD+. Okay. That gets us our thioester functional group. Now we can have water cleave out the cysteine, and we now have our nucleophile that can hook up with our phosphate. Okay? So we're doing some kind of crazy chemistry within that, using that thiol to help facilitate all of that electron transfer. Kind of a complex little mechanism going on there. We'll see other cases of what's happening further on down the road. After we've put the second phosphate on, now we can immediately remove it in our very next step. But in the process of removing it, I have to dump that phosphate onto something else. A very good something else to dump that onto, ADP. Now I've got my ATP as an energy source, and I'm starting to actually up the energy output out of this whole process. Because at this point, we haven't really done a whole lot of actually producing energy. We've just consumed a lot of energy. Okay. The enzyme in this process is, again, a kinase. Remember what we were talking about with our kinases? Kinases transfer phosphates. Okay, so we're looking at a transfer of a phosphate, technically PO3, but we can call it 3 or 4, depending on how you want to look at it, to or from ADP, ATP. Okay, so depending on which direction you're going, it's still a kinase because you're still transferring that phosphate group. It just depends which way you're going. The end result is we end up with 3-phosphoglycerate, which can then move on into our next reaction. An important term that pops up out of this reaction actually is this substrate level phosphorylation. We are producing ATP, so a big energy source or energy currency is coming out of this. But our standard pathway for ATP is going through oxidative phosphorylation. So that's using the electron transport chain, generate proton gradients, which we'll talk about later, to generate ATP. Okay? This is not that process. Okay? So we have to come up with a new name for it. Here they're going to reference it as the substrate level phosphorylation. So we're not using an oxidative process, we're using the, for lack of a better word, a substrate, some other chemical structure to transfer that phosphate group around as an intermediate. The next part of this is we need to transfer that phosphate group. Okay? So we're going to shift the phosphate from our terminal position to the internal one. And what this is going to do is set up an elimination reaction, which will be a lot easier if we can eliminate the oxygen off the bottom of our structure. Okay, if we can eliminate that OH. That elimination reaction is faster in that case. Okay, so we have to get that phosphate group shifted upwards. To do that, we need a new enzyme. The relationship between these two structures is ultimately isomers, so we might consider using an enzyme called an isomerase, except the isomerase enzyme was shifting a carbonyl compound around. Okay? So in this case, yes, we're looking at isomers, but we're not doing the same thing as an isomerase would do. So that's where our mutase comes in. For lack of a better description, we're mutating our molecule somewhat by just picking up an arm and moving it to the leg. Okay? Yeah. The next part of this is we are still, again, trying to end up with our pyruvate structure. So something that might actually be helpful to have on here would be our structure of our pyruvate. Let's draw that real quickly. CH3, OH. We've got our pyruvate structure. Okay, so we're starting to get closer to it. We've got our oxygens in the proper positions, but we don't have just the straight up methyl group at the bottom. Okay? So we have to somehow change the oxidation state about that position. So what we'll end up going through and doing is taking advantage of the enol chemistry. So enol chemistry keeps popping up in this. Okay? We can go through and do a simple elimination where we take the electrons away from that hydrogen, and we can then kick out the leaving group, which will ultimately be made better, of our hydroxide to make our new pi bond. This reaction, even though it's an elimination, which tends to be endothermic, is resonance stabilized with that oxygen. Okay? So that's one of the reasons why this elimination does work fairly well, is we have that resonance stabilization. And it's also going to put an alkene in there that's going to be a little bit more active than if we had done the elimination in the previous step. Okay. So in our last step, what we need to do is have that alkene pick up a hydrogen. Come on. We need that extra hydrogen to get to our pyruvate, and we need to lose the phosphate group. Okay. Well, our phosphate group could easily be picked up by our ADP. Okay. Our oxygen would then become negatively charged, and what we have at that point would be an enolate. So instead of going to the enolate, what can happen is our electrons can shift all the way down. These electrons come out, pick up an H+, and we would have formed that final bond. And now what we end up with is our pyruvate. Okay. Again, what we're transferring across in this instance is a phosphate group, so what we're looking at is, again, a kinase for this reaction. Okay. And we end up generating our second molecule of ATP and our pyruvate molecule. So at this point, we've consumed 2 and produced 2 that's not really getting any positive energy. Where's our positive energy coming from? Now, our electron transport chain doesn't have to do with this yet. This process still produces energy. What did we start with? We started with glucose, which had how many carbons? 6. How many carbons are shown here? 3. So we've consumed 2 and produced 2, but remember the pyruvate reaction happens twice because we cleaved glucose into 2 pieces, 2 glyceraldehyde phosphates. So as we go through that process, converting 1 glyceraldehyde into our phosphate, okay, we produce 2 ATP. But remember, there's 2 glyceraldehydes involved in this process, okay, or produced in our glycolysis of a single molecule of glucose, which means the second one produces our surplus. Okay? So, okay, there we go. Your textbook makes reference to a couple control points, which they've given kind of nicely in these stars. And so these are our big control points. Where'd it go? Within this process. Okay. And in each of those control points, or not each of them, but at certain aspects you'll notice that certain things are going to be contributing. So you'll notice that 2 of these are inhibited by ATP. Why would ATP inhibit these reactions? Not quite. The whole point of this process was to do what? To produce energy, to get that surplus of 2 ATP. Why would ATP inhibit this pathway?
So it can stop producing ATP.
Stop producing ATP. Okay? We only want enough ATP floating around to do the reactions that we need. So if we're at a surplus of ATP, there's no reason to go through and do glycolysis. So we want to shut down that process because all we're going to end up doing is producing more ATP that we can't do anything with. That high energy molecule could potentially go out and start reacting with other things in solution, activating different enzymes, and we don't want that to happen. So we want to minimize that presence. We want to keep it as a relative balance, almost like a buffer system. Okay? But what happens is our ATP levels drop. Well, now those reactions need to be activated because those are going to start producing ATP so we can get our ATP level back up. Okay? So ATP is an important molecule as far as eliminating those or limiting those reactions. We've got a limiting factor right up at the beginning. Why right at the beginning? As soon as we start this process, we're going to start trickling into the energy production system. If we inhibit right away, this glucose can be cycled over to other mechanisms. For instance, storage. Okay, there's no point in starting to process it through our glycolysis method if we don't need that energy. Okay, so one of the things that will inhibit that is our glucose 6-phosphate, which happens to be the product of that reaction. So if we have an excess of that product, we don't want to start producing more of it. Again, we're trying to maintain a balance, just like we did with the buffer system. Okay. There are a couple other enzymes that kind of jump in the way and contribute to how each of these mechanisms end up working. Okay. There is a section in your textbook which I would highly recommend you go through and read because it's a question that's asked in the sapling homework. Okay? And it's looking at what are the different contributing factors. Okay? Some of the more important, relevant contributing factors would be substrates. Okay? Anything being involved in that individual step can potentially inhibit or activate that reaction or that enzyme to start working. Okay? The less obvious ones are looking at, say, calcium ions. Those are contributors to secondary enzymes, which can then affect these enzymes functioning. Okay? After that, we can then look at what happens to pyruvate. So in this section, we aren't concerned about the aerobic systems. We're concerned about the anaerobic, meaning there's no oxygen present. Okay? In animals, what we end up doing is converting pyruvate over into lactic acid. Okay, this is done through the lactate dehydrogenase enzyme. Kind of an odd term with that because it says dehydrogenase, which means to you, what does D mean? Remove, and hydro.
Water.
Look at water, it's actually referring to just hydrogen in this case. What's happening as we go from pyruvate to lactate? You're adding hydrogen, not removing hydrogen. Why was it called dehydrogenase? When they first discovered it, when did they discover it? They discovered it in the reverse. That's why it was called a dehydrogenase. Right? Kind of an odd little tidbit. So some of the names kind of make sense. Realize that you're looking at equilibrium. The enzyme can catalyze both directions. Just so happens in this case what they saw initially was the reverse reaction, so they called it a dehydrogenase. Okay, so don't put a lot of weight in the names of the enzymes, but it does kind of give you a tip-off as to what's happening within it. Okay, we are looking at the exchange of hydrogen through this reaction. Okay? To do that, if we again look at just our pyruvate, okay, we're putting a hydrogen here and a hydrogen here. What charges would those hydrogens ultimately be? Or what charges should those hydrogens be to cause this reaction to occur? What charge do we typically see for hydrogen? Positive. So the red hydrogen makes sense for it to be positive because oxygen is highly electronegative, which means it's probably sucking electrons from our carbon. That oxygen is becoming negative. So the red hydrogen is almost undoubtedly a positive hydrogen. Okay? But what happens with our carbon?
It needs to be negative, it's bringing in electrons.
Our hydride needs to be negative because our carbon is positive. For the same reason our oxygen is negative, the carbon is positive. Hydride, our blue hydrogen, has to be negatively charged to facilitate this reaction. Well, where do we get a negatively charged hydrogen? NADH. So to facilitate the dehydrogenase mechanism, you need NADH. Okay, because that NADH, or the H of NADH, is our H minus, which is what we need to dump in there. Where's the positive hydrogen coming from? This is always frustrating for me. Somewhere in solution. Okay, and we'll get into, and when we move into the Krebs cycle, comes from the enzyme. Where's the enzyme get it? Somewhere in solution. Yay, okay. Remember, we're in a buffer system. We have a relatively constant concentration of H+. We can pull hydrogens from water if we need be. And if that's the case, we consume water. So water, we have a kind of an unending supply of hydrogens. Okay, so we don't want to be too concerned about the H pluses and how they're shifting around. Just realize they are freely available within our samples. The dehydrogenase, that one is another tetramer. There's two common subunits that go into this, the H and the M. H stands for heart, and M stands for muscle. So just like we had, I already forgot which one it was. I think it was was it phosphofructokinase? You remember where we had L and M? I think it was the phosphofructokinase. We had L and M for liver and muscle. Okay, and we said we could come up with different isozymes. Cool, it's always nice when I know I'm right. So rarely happens. Okay, we get different isozymes. We're going to end up with the same issue here. We're going to get different isozymes. So if we took heart muscle, which is something that we could theoretically do in a biochem lab, and grind it up, we could isolate the lactate dehydrogenase out of the heart tissue. And if we evaluated and found out what things made it up and compared that to lactate dehydrogenase that we got out of, say, any other muscle, we would notice that they have different characteristics. In particular, they have different masses. Right? And it's because they're trying to do different processes, or slightly different processes. So we're going to see different systems come out of that. I don't think I quite wanted to get to that. Why are we concerned about running this reaction? Why do we need this reaction to be happening? Okay, why have a lack of oxygen mechanism? How is it still producing energy? If anything, this is taking a very high energy NADH and it's getting rid of it and turning it to NAD+.
Using energy storage.
It's not doing energy storage necessarily either. Remember, ATP is our primary former. Okay? If you go back and look at all the individual steps of glycolysis and our pyruvate conversion, there are several steps within that that require NAD+. So if we go through this process, we've now constantly formed out a bunch of NADH. If we now no longer have oxygen, we can't go through the Krebs cycle and deal with our aerobic consumption. So what ends up happening? We're now going to stall out and not be able to produce any energy. Well, if we don't produce any energy, what happens? The cell dies. Okay? So we want to still be able to produce energy somehow. What we need is more NAD+, to facilitate the earlier steps of our glycolysis mechanism. Okay?
When we don't have any oxygen present, don't we just switch cycles and start going through alcoholic fermentation? Oh, okay.
Okay, alcoholic fermentation or lactic acid. Okay? We don't go through alcoholic fermentation, we go through the lactic acid cycle. Okay? But there are other cells that go through alcoholic fermentation. Okay? And that's what's happening. The whole reason that we go through and do that is to generate that NAD+, because that NAD+ can then be used with glyceraldehyde phosphate to generate our BPG, which can then generate ATP. Okay, so we can almost get a trickle current of ATP as long as we constantly have NAD+. Okay, if we go through this process and just have ATP and NADH, we can't produce ATP anymore. Okay, if we shut off every other reaction afterwards. So the lactate dehydrogenase, or your alcohol, our pyruvate dehydrogenase, they kind of showed this oddly, are involved in producing that NAD+. Okay, once we have that NAD+, we can cycle off the rest of it. Okay. So in alcoholic fermentation, you get a series of two reactions. You're decarboxylating pyruvate to acetaldehyde, and you're doing acetaldehyde to ethanol. Okay, the enzyme involved in this is a decarboxylase. This is kind of an interesting reaction because it requires the presence of thiamine pyrophosphate. Okay, kind of a fun process because one of the things we like, or I like to say about beer, is that you've got yeast cells in your beer. Well, your yeast cells require the presence of vitamin B or thiamine. So if you're drinking beer, you're getting your vitamins, your daily nutrients, because you need thiamine to cause these reactions to function. Okay, so that's why we drink it, for the health reasons. Okay, and then we also have the dehydrogenase that catalyzes the final step. There are studies that actually prove that one beer is very beneficial, okay, particularly if you're dehydrated. You get lots of electrolytes in that beer, lots of extra vitamins. As soon as you pass that one beer threshold, you've ruined all of those benefits. Okay, so if you go for a long run, one beer helps you out, then start drinking water. So, thiamine. We're going to come back to this in a couple different ways because it shows up now and it shows up in the Krebs cycle, and I was, however you want to look at it, either too lazy or too busy to go through and condense these two sections into one thing. So if we go through and take a look at our thiamine, okay, really the important aspect is nicely highlighted by your textbook in that red ring. Okay? That ring is what's really driving the chemistry in this. Okay? Kind of a neat process. Turns out, if we go back to what I've tagged here, we actually have a relatively acidic thiamine structure. Okay? Where is that acidic hydrogen? And as a hint, it's in the red part. Or it's sort of in the red part, I guess. It's another hint. It's now in the red part. Okay. That red hydrogen is actually relatively acidic. Okay. If we go back and look at this, and it's kind of hard to see it on this figure, but that nitrogen is positively charged. So what is that nitrogen doing? Sucking electrons away from the carbon, which means what is the carbon doing? sucking electrons away from the hydrogen. Okay? So what ends up happening is that carbon picks up a negative charge, and we now have a relatively strong nucleophile. If we take this actually a step further, what we have is both a nucleophile and an electrophile. Okay? Because the electrons that the carbon is getting from the hydrogen are in a perpendicular orbital, and I know everybody loves this discussion, in a perpendicular orbital to the p orbital of our pi bond. Okay? Very rare for anybody to talk about this in organic chemistry, but we'll see it pop up in a second. What we have formed at that carbon is a structure known as a carbene. Whoa, that's an eraser. That carbon is officially neutral charged. Okay? But it's a really weird looking structure. Okay? That just doesn't make a whole lot of sense. What's actually happening within this is if we pull this structure, right now it's kind of in a planar geometry. It's sp2 hybridized because of the 3 groups of electrons. Well, if it's sp2 hybridized, what's left over? And not sp3, that's changing the hybridization, a p orbital. There's a leftover p orbital which is currently coming in and out of the plane at us. So if we pulled this structure kind of up out of the board, and this is kind of weird to show this, but we've got the lone pair kind of coming at you and the other bonds. Our p orbital is above and below the plane, which means this carbon has a source of electrons, nucleophile. It also has an empty orbital, electrophile. Okay? So it can do a lot of kind of interesting, bizarre, crazy chemistry because of this characteristics, because of these characteristics. Okay? Our initial process is forming that negatively charged actually, before I even do that, I think I got the mechanism there. Yeah. Okay? We can form that negatively charged carbon up on our thymine. Once we have the negatively charged carbon, that can then attack a carbonyl. Okay? In this case, they've kind of glossed out what all those R groups are. So let's go back real quickly and take a look at pyruvate as a structure. With pyruvate, how many carbonyls do we have? We've got 2 carbonyls. Which of these 2 carbonyls is our electrophilic one? Which one is the one they're showing in the mechanism is reacting with thiamine? Okay, why the top? Okay, we have 2 oxygens attached, and this is a very common thought process. Electronegative oxygens withdrawing electron density. If you go all the way back to organic chemistry and we think about the relative importance, okay, if we think about acid-base strength, we start with looking at stability of charge. Then we look at atom effects, and there's 2 atom effects that we evaluate. One is size, one is electronegativity. Which one trumps? Size, not electronegativity. Why is that relevant? If we then push to a molecular effect, we're doing the same thing, size and electronegativity. Okay? But in the molecular effect, we call it something else. Our size is resonance, and our electronegativity becomes an inductive effect. So what you are referencing by saying this top carbonyl is more electrophilic is you are saying, based on an inductive effect, there are more oxygens around it. But before you look at an inductive effect, you must look at a size effect or resonance. What does that do to the carbon? There is resonance stabilization of that carbonyl carbon, which means that carbon is no longer as positive, which means the bottom one is actually our electrophile. Okay? We can think about this another way. If you look at the reactivity, sorry, my stupid pen. Okay, and this is for those of you that took second semester organic, because I don't think you saw this in first semester. Two functional groups, right? Carboxylic acid and a ketone. How does the carboxylic acid act? As an acid. How does the ketone act? As an electrophile. Okay. If the inductive effect was important, guess what our carboxylic acid would be acting as? An electrophile not an acid. Okay? So it's a resonance effect that stabilizes that carbon. Can we make it act as an electrophile? That's absolutely true. But that's relative to those two functional groups. Our ketone is more reactive. Right? So when we go through and look at this mechanism, what ends up happening is our thymine attacks that carbonyl. We see a whole bunch of acid-base transfers, lots of decarboxylation kind of steps. So if we draw out the rest of that R group for pyruvate, R1 is actually the carbon oxygen. So what we do in that elimination is we are now kicking out this group, also known as CO2. When we look at alcoholic fermentation, okay, what's happening with that CO2 production? I guess if anybody's brewed, you might know this. If you haven't brewed, then maybe you don't. Okay. Well, we go through two stages of alcoholic fermentation in brewing. One is to produce the alcohol, but we don't cork it, okay, because we want more alcohol in it. Eventually, we take that sample and we put it into a bottle and we cap the bottle. Okay, now what happens? It's still fermenting and it's producing CO2. Now what does the CO2 do? Act as a toxic gas and kill the yeast. So that secondary fermentation is not to produce alcohol, but to produce CO2 to give us carbonated alcoholic beverages, okay, and dead yeast cells with all of our nice vitamins. Right? So it can continue to go through the reaction. Ultimately, you end up, and the arrow pushing on this one was a little bit weird, you end up at the exact same place that you started. Okay? Your thymine structure with that negative carbon. Okay, so it's kind of a neat process. You look at each of those individual steps. Okay, what's happening in each of those? Kind of makes sense? Okay, I personally find this absolutely fascinating. You're bringing in a crazy organic molecule to do crazy bizarre chemistry we never would have considered. Okay, also probably one of the reasons why biologists say they don't want to look at it, because all they care is does it work. Okay, forest versus the trees argument. I'm looking at the trees, biologists are looking at the forest. Okay? So we're both important, but it comes down to which we see is more relevant. You could make the argument, I'm not looking at the forest, I'm looking at the bark. Or sorry, the tree, I'm looking at the bark. But that's neither here nor there. So if we look at our overall process, glycolysis is an exergonic reaction, net, and we're going to produce roughly 73.4 kilojoules per mole. Okay? In this net process, we're also going to produce 2 moles of ATP as an overall result. Okay? Because I know we all love to memorize numbers. Anybody remember the delta G for ADP to ATP? Anybody remember the delta G to go from ADP to ATP?
The difference between the two?
It's your delta G for the reaction.
The exact delta G?
Negative 30.5.
Delta G ends up being for ADP to ATP. So the first thing we should focus on, should that be a positive or a negative?
Should be a positive.
It should be a positive. We're adding energy into that. Okay. So the value you're probably looking up where you said negative 31 was ATP to ADP. It's actually going to be a positive, what was it, 31?
30.5.
Everybody comes with different numbers on this too. They're all ballpark 30. 30.5? Okay. Why is that kind of important? How much energy was our exothermic reaction? 73. To convert 2 ATP across, how much energy do I need? Double that. I need 60, what is it, kilojoules per mole. How much energy did I produce? 74. Okay. So it's not a perfect transition across, but if you go back to physics and thermodynamics and all that fun stuff, we can't have a perfect transition. Okay. But we have an exothermic enough reaction to provide enough energy to produce 2 ATP. Okay, so that's ultimately all that's really kind of going through with this summary and looking at those delta Gs. Okay, questions on that? I'm pretty sure that is the last slide for 17. So are there any questions from 17?
We can email those to you, right?
No, I'll never answer questions. Yeah, absolutely. Email works. I am still holding office hours today too. And I will be holding office hours actually Friday, which I need to tell everybody else about too, 12 to 2. So I will be in the tutoring center Friday as well, 12 to 2. Okay? So, writing center.
You're what?
They'll be in the writing
Oh,
center,
the writing center?
writing the paper.
Oh, the paper that's.
So, chapter 19, Krebs cycle. So we're going to be doing kind of a similar process. We've got our overall reaction. So they gave us a nice kind of figure showing all the different pathways that can connect through and all the different energies and electrons and all sorts of other fun stuff. Okay. What we're looking at, I keep referring to it as the Krebs cycle, which I guess I should stop referring to it, but tough, is the citric acid cycle. So what ends up happening is our glycolysis process goes through and produces pyruvate. We convert the pyruvate to acetyl-CoA. Acetyl-CoA then shifts into the citric acid cycle. Why is the acetyl-CoA thing such a massive looking piece? I'm making stuff up here as I go too, by the way. Why would we make that such a massive looking piece?
It facilitates 3 different reactions.
So it's being produced from 3 different reactions. Okay, another thing that I think is kind of important on why it's such a big piece is that is a massive enzyme complex. Okay, it is huge. You're running 3 different, effectively not even mechanistic. You're looking at 3 enzymes all coming together in kind of one piece to produce a bunch of chemical changes. Okay, so that is again, we come back to this whole forest through the trees thing. If we look at our citric acid cycle, so we look in this method, acetyl-CoA, so pyruvate to acetyl-CoA to citric acid, big old chunk. If we look at how it's typically shown, what just changed? I don't know what just changed there, so let's just advance. All right, we can now look at the Krebs cycle. We got everything in our Krebs cycle. Where's our conversion from pyruvate to acetyl-CoA? Tiny little space. Tiny little space. Okay. Why is it now so tiny?
Because it's at the very beginning of the process.
Why is it at the very beginning? It's only one step. Krebs cycle has a whole bunch of different steps. So why was it so big on the other side? Okay. It's because there's a lot of stuff happening in that tiny little step. Okay. So what we're doing is taking pyruvate to acetyl-CoA, and I'll try and draw those, actually they're there, but I'll draw your attention to them. The process of doing that, you need coenzyme A, you need NAD+, and you need a bunch of other stuff that isn't actually listed in this. Awesome. Okay. Those bunch of other things are provided by the enzyme. Okay, so the enzyme is facilitating a lot of those changes of bonds. So where is our pyruvate? So if you remember the structure of pyruvate, okay, if we look at this diagram, I know it's kind of blurry. What we're looking at is our black being our carbons, our red being our oxygens. So our pyruvate is right up there at the top. We've got our 3 carbons, our oxygen, and another oxygen. So we've got our carbonyl. Man, that red over red doesn't work. Is that a little better? And our carboxylic acid. So we've got our acid functional group out here. Okay. What's happening as we move to acetyl-CoA? Well, where's the acetyl-CoA? Carbon, carbon, oxygen. Yeah, it's kind of coming in over the top. And then our sulfur-CoA. Right, so we pull that out a little bit. What changed? Let's erase that, make that a little cleaner. What changed as we went from, I'm going to remove hydrogens from our oxygens, as we went from pyruvate to acetyl-CoA? And if it helps.
Sulfur came in and was going to break that compound in half.
So our compound is kind of breaking in half, if you will. We are cleaving that bond. Okay, and we are replacing it with the sulfur-CoA. What function, or what bond, or what piece is that? Almost, but if I cleave it from the structure, it becomes, it's a carbon and 2 oxygens, it's CO2, it's carbon dioxide. Okay. So we are doing a decarboxylation reaction in this process, okay, because we have to lose that CO2 from the structure. Okay. Why does that become a little bit problematic? Well, if we use arrows, we're now putting extra electrons on that carbon, and that's really hard to do. Okay? So that's where the enzyme is going to come through and facilitate that electron transfer. We saw this already in our lactate dehydrogenase, I believe it was, okay, where thymine came in and helped out and facilitated the CO2. Okay? Well, we're doing the same thing with this enzyme as well, but it's going to involve a lot more pieces, because instead of producing CO2, I forgot what the other piece was. What was the other piece? CO2 and, acetaldehyde. Okay. We need to actually make sure that the carbon in that piece is still receptive to chemistry, okay, that we can then transfer. So what we need to do is transfer an acetyl group. That carbon needs to maintain a solid positive. So we need something attached to that carbon that is going to come on and off relatively easily. That's our our coenzyme A, okay, with that thiol group. Yes.
In the book or whatever, it's not really showing. I don't think it's really showing the specific chemistry of the. But it's showing that it accepts the electron and then facilitates that.
That's
so.
what we're going to look at here in a second. So I personally get frustrated by that, because I look at that and I say, how the heck is the chemistry happening? There's way too many electrons floating around. That's just nuts. Okay? So if we push that further, we can go out to something like this. I don't remember if I got this from your textbook or not. I may have gotten it from the internet. You might see, no, it's there. So we're starting with our pyruvate. And we are ending with our acetyl-CoA. Okay, so that one single nice little arrow that they gave us to begin with is now actually a combination of all of these possible arrows, okay, showing each of those intricate intermediate steps. And you'll notice that one of the very first thing that pops up, the thiamine. Okay, that thiamine is, again, our important molecule that's doing a lot of the electron transfer, or a lot of the bond exchanges to allow this process to occur. But not only is it thiamine, you're now also dropping in this molecule, which is lipoic acid. Okay? It's in reference to all of the carbons, and then your 2-thiol, officially here, 2-thiol groups. What's happening as we go from the 2 thiols to the disulfide bond?
Creating this structure.
Yeah, we have a
I
ring.
want to call it a single structure.
Okay, let's focus on just what happened beyond that. We start with, we remove hydrogen, which means we did, so we could do looking at a dehydrogenase, hence our enzyme name. Okay, it's a oxidation. Oxidation, because we've lost hydrogens. Okay. So we are doing a lot of reduction and oxidation throughout this process, and it is very misleading, to me at least, to show this just as a single arrow as a lead up into the Krebs cycle. There's a lot of stuff going on within this. Okay. This is where, again, the biologists say, I don't care, it does it. Forest through the trees. Okay. I'm more concerned about the forest, biologists are looking at the trees. Or other way around.
Reverse.
Other way around. Maybe I like looking at forests. I'm looking at the trees, and biologists are looking at our forest. Okay? So we want to be aware of what's happening within that. We may not need to understand it perfectly, but we need to know it's a little bit more complex than just, oh, a reaction happens. Okay? So this is now looking back at the actual mechanism of our carbene formation. Okay. The net result of this reaction is we end up with this guy. Okay. Really, really crazy. What happened? The bulky base reacted with this guy, and we end up forming this molecule, which can then attack the double bond, and the double bond can attack it. And that's how we end up with our product. So that's our carbene molecule. Okay, so this happens to be one way that we can go through and make it. We have a source of electrons, and we have an empty orbital in which we can dump the electrons on our carbon. Thymine does the exact same thing. Okay, we've got a slightly different structure, or a slightly different orientation of it in this case. Okay, our carbon with the acidic hydrogen. If I remove the hydrogen, the carbon now has a lone pair of electrons. The nitrogen, it's nice of this figure to actually have the charges on it. The nitrogen is formally positive, so if I erase that hydrogen and have that negative charge on our carbon, but because the nitrogen is positive, it can suck electrons away. That carbon is now both a source of electrons and a receiver of electrons. Both a nucleophile and an electrophile, which is exactly what happened with our carbene. So it's kind of a neat process, because you might think with the carbene, if you saw it in organic chemistry, there's no way in hell this would ever exist on its own. Holy crap, biochemistry already did it for us. Okay. So this molecule is going through and generating that exact same process, but in a much more stable situation. Okay. It's only reactive when it's put into this kind of enzyme process. Kind of a neat little deal. Okay. So I think it's neat because we have a stable carbene. We've got another version of the mechanism which actually shows it in a little bit more detail. Okay. When we looked at the dehydrogenase, our primary difference comes in. I'm going to back through it and find it. Okay, so our carbene comes in attacks. We can decarboxylate. We can pick up our hydrogen. We can do the elimination. So we've got our enol. Okay, and this was our primary difference right here. Okay, it's this step. Okay, this step going right here is the big difference, okay, between our anaerobic process and our aerobic process. In our anaerobic process, those electrons came out and picked up a hydrogen. Okay. If we're now trying to make acetyl-CoA, those electrons are going to come out and attack the sulfur of our lipoic acid, which can then pick up a hydrogen. Okay. That generates this fun little intermediate, okay, which can then go through, reform our carbonyl again. It's kind of nice about having the thiamine. And now when we go through, we can have a really good leaving group with our lipoic acid. Acetyl-CoA now has the opportunity to come in and attack, which ultimately kicks out our lipoic acid, and we end up with our acetyl-CoA. Okay. Very neat, fascinating reaction, if you ask me. Lots of intricate pieces happening within it. A lot of those pieces are being supplied by the enzyme itself, or that enzyme complex. Once it picks up the acetyl-CoA, this can then go on to go into the Krebs cycle, because acetyl-CoA goes into that. But our enzyme is not recharged yet. We've pretty much taken everything out of the enzyme. It's now lost all of its reactivity. So we have to refresh it. Okay? That refreshing process has to come back through FAD. FAD, being a big molecule, is kind of locked within the enzyme, can't really move around. So what ends up happening with the FAD, it accepts the electrons to get us back into our lipoic structure. Once we now have that back, we have FAD, where to go? FADH2, but that can't leave the structure. So FADH2 is now freezing up and holding up the system. So we need a small electron transfer carrier molecule. What is our small electron carrier? Yeah, we have a smaller, or at least a smaller one than FAD. NAD+. So the NAD+ will then pick up the hydrogens from our FAD And we've now transferred those electrons fully out, and the NADH can now go out into its reaction. So if we go all the way back to what's happening within our reaction, we need the acetyl-CoA to come through and get us, we need the coenzyme A to get us to acetyl-CoA. We also need the NAD plus to form NADH. Why do we not mention FAD? Because FAD is not mobile. It's a prosthetic group within the enzyme. Okay, so it can't come out of the solution. NAD is the one that comes in, picks up those hydrogens, and ultimately clears it from the enzyme to reset it back all the way to the beginning.
So the FAD is inside the
It's
coenzyme?
enzyme, inside, not coenzyme A, it's in the enzyme.
In the enzyme
It's
itself.
in pyruvate dehydrogenase.
Oh, gotcha.
Yeah, you got all these tiny little pieces. Okay. So the other thing that I like doing is looking at the image of some of these. So we're also going to pull up, as soon as I clear all that, step away from those. We can take a look at this guy. Let's see. What? Oh. Okay. So this is something that I've shown before, and I don't know if people realize what it was or how useful it is. This is the PDB, the Protein Data Bank. So a lot of you are doing writing assignments on individual proteins or what those proteins are being affected by. You can go into the PDB and type in the name of your enzyme, and it can potentially pull up the enzyme structure. So what we have in this case was our, which one did I look up? Our pyruvate decarboxylase structure. And what we're looking at here is that whole enzyme, or part of that enzyme, because it's that big. But you can see that we've got nice little channels running through it. Okay. Oh, no, let go of that. Okay. So we can see channels, we can see openings on how we might sneak into it. You'll notice that we've got these other small little groups in there. Those small little groups happen to be ligands within the enzyme. So they would almost be a prosthetic group to our enzyme. Okay. So we can rotate and change this structure around to see what's going on with it. If you're looking at a particular drug synthesis, this can be very helpful because you may get lucky, and what someone may have done is found your enzyme that the drug targets and it crystallized it with the drug in it. Okay. You can then change your view to say just look at the ligand, and you can now rotate and move the structure to see what amino acids are interacting with the drug. Pretty neat process. Okay, so the PDB is highly useful. You can see all sorts of fun little things happening within it. You've got your alpha helices, you've got your beta sheets. Okay, in yellow, what's all the white stuff? Just some loops. Okay, it's not necessarily a beta U-turn or anything like that. It's just our amino acid sequence that is now mobile. Okay, that can freely move around at least a little bit. And all of this is coming out of, I think this one is actually X-ray crystallography. Crystal structures, so we're looking at X-ray crystallography in this one.
The little white lines, can you say that those are primary structure? Because they're really not doing anything? They're just
Not really. So primary structure really is kind of a misnomer. It's not a structure.
It's just sequence.
It's just the sequence of amino
So it
acids.
would be secondary?
So this is more secondary structure, and it's not even really secondary because secondary is, how do we want to say this? The arrangement of your primary structure into organized shapes. Okay. Did I say sheeps? Into organized shapes. Okay. Those organized shapes are your alpha helices and your beta sheets. So this, the white lines aren't really secondary structure because they aren't organized shapes. Okay? They aren't primary structure because the primary structure is just the sequence of amino acids. So how those orient in space is now your tertiary structure. And it's not organized, so you can't call it secondary structure. Okay? Where's my mouse? Okay. So kind of a neat tool. If you want to use that for your presentations, you can. It's not that big of a deal. I think it's helpful. Okay. And so this is the biggest conclusion that I got out of biochem, which is one of the reasons why I'm less of a fan of it, even though it does really cool stuff. But ultimately, when we talk about biochem, the conclusion is if we want something done, the enzyme does it. Okay? How did this process do it? I don't know, the enzyme did it. Right? It's true, it works, and depending on what you're trying to do, that's all you really need. Right? I just want to see more than that as a personal preference, so I tend to go in and look at all those mechanisms and arrow pushings. So possible exam questions that could come out of this. What role does thymine play in many redox reactions? That one's kind of tricky. So what did thymine do?
Well,
It stole electrons.
yeah, so
It's A and B. Its first action in this case was acting as a nucleophile.
But then-
After it acted
then
as a
it's
nucleophile, then it acted as an electrophile. So you end up doing both.
about that, so would
So
you
A and B.
want, would it
I would
be
edit that
A,
question.
okay.
Yeah, I'd fix that.
Because I was like, wait.
Thymine is an example of a naturally stable.
I could radicalize,
Carbene.
right?
No.
But I still
Yeah,
don't know.
not easily. Does the conversion of ADP to ATP require oxidation?
Yes.
No.
Oh, no, it just.
It does not.
Energy.
All it requires is that energy. You're not doing a redox reaction necessarily when converting to ATP. And in most of the cases that we've looked at, even through glycolysis, it's not
That
there.
comes after.
That comes either before or after. The actual conversion to ATP does not require oxidation. Okay. Which of the following does not contain enolizable hydrogens? So, enolizable, what do we need? Let's break down the name. Ene and all. What does ene mean?
Alkene.
An alkene.
And OL means alcohol.
And alcohol, or sorry, all means alcohol.
I like that epoxy ring.
So what we've got to go through and do is convert these structures into a situation where we have an alcohol and an alkene. Okay? So our enolizable hydrogens means we remove a hydrogen and we can now make an alkene and an alcohol. Okay? The only one that can't do that, or actually, oh, this isn't even multiple choice, that's mean. So, where are their hydrogens? There's hydrogen there. What happens if I remove that hydrogen? I end up with negative. What's the resonance structure?
Yeah, you're not creating an alkene. You're not, alkane.
We have the alkene. We don't officially have the alcohol, but what happens if I put a hydrogen on there? Okay, we do have our alcohol. We have enolizable hydrogens. So what we're looking at is that position. Okay, I'm actually going to go ahead and make the statement of no.
The position of what? No, the position of
Okay?
what?
Not just because we haven't talked about it in enough detail to reference that. And the last time we brought this up, almost everybody had gone through second semester organic. So go ahead.
Position of what though? Position, the carbon position, the alkene? See what I'm saying? You formed an alkene at the carbon. But are you talking about at the carbonyl carbon? I don't understand. When you're talking about position, you just formed a
Position.
new double bond and removed. a
So
double
enolizable hydrogen means you're removing a hydrogen that now puts you in a situation where you have an enol.
bond. Okay,
If we remove a hydrogen, we have an enol.
okay.
Okay. And we haven't gone through that in enough detail to worry about it, so we'll cut that question out. Why might it be important for citrates? Why is this question showing up already? That question shouldn't have shown up yet. I must have shuffled the enzyme or the questions around a little bit. Sorry about that. So we'll skip that question for the moment too because we haven't talked about it yet. Citric acid cycle. So when we move into the citric acid cycle, we've now got a summary of all of our individual reactions, okay, and their enzyme names associated with each of these. Okay. Again, this is something in a standard biochemistry class. You should know every last little bit of this. Okay. You should know a lot of it in this case because we're not going through each of the individual reactions in detail. So that means you're going to be responsible for knowing those names and those enzyme structures. Or not the structures of the enzyme, the structures of the molecules. So, if we kill that Krebs cycle drawing that we had initially with all the colors and the bonds and the enzymes and all that fun jazz, and simplify it down, this is what we end up with. Okay? So we're just looking at each of the individual molecules within the Krebs cycle itself and how we're converting in between these, or addressing potentially how we're converting between these. Okay? Once we've got our acetyl-CoA, which we just talked about, we can then react the acetyl-CoA with oxaloacetate. In the process of that, we make our citrate. Okay? So what's happening in that reaction? And this is something I fully expect you to be able to do. What I would probably ask is what things changed. Where did the acetyl-CoA come, or where did the acetyl group go? Where's oxaloacetate? What we want to do is compare across and see where we've got similarities.
Acetyl-CoA attack the oxaloacetate.
Yes.
Right there in the middle.
There in the middle.
Point. Actually, no, it wouldn't. Yeah, wait.
Okay, so let's break this down. Let's find oxaloacetate.
But it's the.
Where's that group in oxaloacetate?
Right in the middle. It's the. Oh, wait.
What group are you asking this on the middle?
The red dot. Where's the red dot in citrate? I know, this is not easy. Okay, I agree. I think it's on the left too. So where's the next carbon?
Oh, yeah, it is. It's the next one down.
The next carbon.
Yeah, there you go. That's where it's at.
All right, now the trick. The last carbon. It's up. Okay. Where is the acetyl group, or the carbonyl of our acetyl-CoA? It's on the right. Where's that carbon? What did that carbon act as in this reaction? Where did it form a bond? So then what we want to do is identify the new bond. The new bond is right there. So that carbon had to form a bond at that connection. So if we went through and now erased all the carbons that are not involved in the reaction, or the dots, what we are looking at doing is connecting the red dot to the blue dot. What charges would each of those atoms pick up? So if we look at the blue dot, we got carbons and hydrogens. Would we expect much of a charge? No, so that one's not easy to see. If we look at the red dot, what charge do we expect? So it is overall neutral, but let's push it. Can we see a charge forming on that? Why positive? Because that oxygen is going to withdraw electron density from it, making that carbon positive, which means, what does our blue carbon need to be?
Negative.
Negative. How can I make that carbon negative? We strip a hydrogen from it, making the our enolate. There's why our enolizable hydrogens are really important to be able to find, because that's why this reaction is working. We have an enolizable hydrogen on our acetyl-CoA. That is what makes that carbon a nucleophile, which can then attack our electrophilic oxaloacetate and push out the reaction. Is it the only thing that occurred?
Where did the oxygen come from?
Is that the only thing that occurred? No, we also have this oxygen popping up. Okay. How did that oxygen come in there?
Actually, I was looking at the,
Okay, we'll come back to that. Water comes in and kicks out our coenzyme A. Okay, so what oxygen were you looking at?
the one on the red dot, in the citrate.
Oh, the alcohol.
Yeah, the alcohol.
Okay, let me before I answer that, can I clear everything? Everybody okay with what's up here?
I'm a little confused with what's going on from the pyruvate. Are we going to, is it reacting with the acetyl-CoA first, or is it going to, because I see like...
Pyruvate forms acetyl-CoA. We already talked about that reaction earlier.
Yeah.
So pyruvate is out of the picture. The only one that we are looking at is there. That's it.
Okay.
Okay. So what ends up happening is the acetyl-CoA has an enolizable hydrogen, which makes the carbon of the acetyl group a nucleophile. That nucleophile can attack our carbonyl carbon here, making the bond between our blue and our red dots. Okay. So this is why identifying enolizable hydrogens is of critical importance to understanding how the reaction works, because almost all of it is enolizable hydrogens. Okay. That creates that first purple linkage. Okay. This last oxygen also somehow changed in this reaction, because if we take a look, that should be a sulfur group. So as a secondary step, water comes in and kicks out the coenzyme A and takes it back to the SH. That's where that oxygen is coming from, is from water. Okay. And now what Diana is asking about, assuming there aren't any other questions with all that mess of stuff up there, which is why I need to erase everything. Okay. I was just going to erase. Oh, let's do it this way.
I see that there are 5 oxygens in the oxalo.
What you were asking about is where did this
Right.
oxygen come from? Okay, well, that oxygen was connected to this carbon. That carbon we said was here. What happens when our acetyl structure attacks that carbon? How many bonds would be at that carbon? Too many, so we have to break a bond. What bond do we break? The pi bond, and we put the electrons onto that oxygen, which is now negatively charged, which can pick up hydrogen from solution. There's your oxygen. Okay? Neat stuff. Okay? Then what happens is we start to move through this reaction. Okay? So we look at, for the most part, we look at enzymes at each of these individual arrows. It's not officially true because citrate to isocitrate is actually one enzyme. But each of those steps, what we're trying to do is see what things changed. So to go from citrate to cis-aconitase, what happened? Okay, we lost the OH. Is that it?
I'm guessing some
Well,
We formed a double bond. To form a double bond, what type of reaction do we need to do? We need to do an elimination reaction. When we do an elimination reaction, we have to eliminate 2 pieces from our structure. So far, we've only eliminated 1. We also lose a hydrogen off of our citrate to do that elimination. What happens when we go from the cis-aconitase to the deisocitrate? We gain an alcohol and gain a hydrogen. We did an addition reaction. So what's your what?
but you're adding water.
No, it just moved the OH over.
Yes. All that happened in the process of doing this is we exchanged the location of the OH. It moved from the middle of our structure towards the side of our structure. Why did that happen? What we're trying to do is activate certain positions within our structure to facilitate other reactions happening. Okay? So the net result on going from citrate to deisocitrate is we just swap those locations. You can't just pick one atom up and move it to the other place. You have to go through intermediate steps. That intermediate step is the cis-aconitase, or cis-aconitase is the enzyme. I don't know what the name of that compound is. Cis-aconitate, maybe? So we have to eliminate first, because what we're trying to do is swap the chemistry of those 2 positions. Once we do the elimination, we now have chemistry still available at both carbons because of the pi bond. We can then do an addition reaction and add back the exact same pieces we removed, but we're going to add them back in a different fashion. Okay? That is what ultimately switches us across to the deisocitrate. What happens when we move from the D-isocitrate to alpha-ketoglutarate?
You lose that carboxylate group.
We lose a carboxylate group, which is probably going to involve what type of an enzyme? A decarboxylase, because we're removing the that one. What else is happening? We're losing, careful, we're not losing a hydrogen,
Electron.
we're losing hydride, because we are oxidizing it. If we lose a hydride from this, we need something to accept that H minus. What would accept that H minus? NAD plus. We're doing a redox reaction, we need a redox mediator. Okay, this is why we talked about those redox mediators to begin with. Alpha-ketoglutarate to succinyl-CoA, what's changing? It's not just adding CoA.
Succinyl-CoA is attaching itself to the carbonyl group.
Which carbonyl? Where is that carbonyl in alpha-ketoglutarate? As a hint.
That one.
It's that one. What does that mean?
It's an electrophile. Succinyl-CoA acts as a nucleophile.
Acetyl-CoA will act as a nucleophile. What else is changing? We're also decarboxylating here. We're losing the CO2. Okay. What happens in the next sequence? Succinyl-CoA to succinate. We lose the acetyl-CoA and we replace it with an oxygen that likely came from water. Why did we not do that in one single step? Because we formed a stable enough intermediate that the enzyme was able to spit it out. Okay, so we need a new enzyme to do that transfer. Succinate to fumarate. What happened? We gained a pi bond, but to gain a pi bond, what do we need to do? An elimination. We need to eliminate two pieces. What are the two pieces we lose? Hydrogen and? Not hydrogen.
Well,
Hydrogen and hydride.
hydride, yeah.
What do we need? We need another electron mediator. Okay. Fumarate to malate. Okay. We're doing an addition reaction. We're gaining the OH and hydrogen. Malate to oxaloacetate. This one's a bit obnoxious. This was unintentional on my part.
You're moving the hydrogen, but gaining a pi bond and moving the oxygen.
There's a lot going on there.
It's actually not that much.
The whole
Well, you're
thing's
losing
rotated.
water. Aren't you losing water?
These two are the same thing.
Right.
What changed? Oops.
The position of the.
We just formed the pi bond. So it's just the position of that alcohol. Or instead of it being an alcohol, it's now a carbonyl. So we're again doing another redox reaction. Okay?
It's switch
So
carbons.
it didn't switch carbons. We took this, so the example I like to give in organic chemistry.
I see. Okay.
What's my
Oh.
name?
Yeah.
Yeah? No? You're just going to give up? Okay, fine. Whatever.
Oh, I see.
No, it's not even that. What's my name? What is it now?
Mike.
Didn't change, right?
Very
What's the relationship between this? It's not a mirror image.
enantiomer.
It's not, you're still going, it's not a mirror image. Did I all of a sudden become a mirror image by turning around?
No, you're not.
It's the identical structure. All that happened is we took this structure and we rotated it to here. I have a feeling that that relationship was just drawn wrong when I copied the structures out, or because I doubt I did that intentionally. And sorry, we are done.